Thank you. Hi. So, during the last few decades, antibodies became a very important class of molecules in biology and in medicine. As you all know, antibodies are in the heart of the immune system and in our ability to fight pathogens, but they are also the fastest growing class of approved drugs and the largest class of molecules in clinical studies. They are so common in biological assays that actually they are the group of reagents on which lab spends most of the money. They are a major tool in diagnostics, they play a key role in vaccination, and more and more. So let me remind you how does this so important molecule looks like. So antibodies are composed of 2 heavy and 2 light chains, where the region on the antibody that binds the antigen is named paratope and is composed of 6 loops, 3 on the heavy chain, named CDR, H1, H2, and H3, and 3 on the light chain, named CDR, L1, L2, and L3. On the antigen side, the region on the antigen surface that binds the antibody is named epitope. So antibodies are very important, but having an antibody is not enough. And we also need to know what exactly does it bind, or what is its epitope. For example, in autoimmunity, an antibody that was raised against a pathogenic epitope binds also a self-epitope. If we want to understand autoimmunity, we need to know what are these pathogenic and self-epitopes. When designing a therapeutic antibody, if we will know what epitope it binds, we will be able to suggest how to improve it and how to improve its affinity and selectivity for its antigen. Furthermore, if we want to submit this therapeutic antibody for approval by the FDA, we need to specify what exactly does it bind. To design a vaccine, we want to know which of the many possible epitopes of the pathogen will cause the production of a broadly neutralizing antibody. To select the most suitable antibody for a biological assay, we need to know where exactly on the antigen does it bind, and so on and so forth. However, this task of mapping antibody epitope is a very difficult task, and practically X-ray crystallography is the only method that can give us the full answer. And this brings us into predicting antibody epitopes. So people were starting to predict antibody epitopes during the beginning of the 80s, where when these methods are trying to predict epitopes, they're actually trying to separate or to identify which residues on the protein surface binds the antibody. Most of these methods were based on amino acid scales, such as hydrophobicity. However, in 2005, Flower and Blythe have published a study where they evaluated the performance of almost 500 such amino acid scales in predicting antibody epitopes. And you can see that each point here corresponds to one such amino acid scale, and you can see that they have a near random performance near the line, which is the random performance in predicting antibody epitopes. Later on, more sophisticated methods were developed, where most of these methods try to separate between epitope and surface residues based on combination of properties of these residues. Some of them also take into account the antigen 3D structure and cluster predicted residues over the antigen 3D structure. But still, last year, Yao and co-workers have published a study showing that many of these methods have an AUC that is not substantially better than a random AUC of 0.5, as you can see here. So altogether, we can say that antibody epitope prediction methods are not accurate enough. But why is that? And we believe that the answer to this question is that epitope residues are basically indistinguishable from surface residues. And I will try to explain this giving the example of amino acid composition. So we and others have shown that the amino acid composition of epitope and surface residues are basically the same. And this is quite surprising because in the case of general protein-protein interfaces, they do have an amino acid composition that is different than that of surface residues. So let me show it in a different way. The x-axis here is the measure for the difference between two sets of amino acid compositions. You can see that the amino acid composition of surface residue and of protein-protein interface are quite different. However, the amino acid composition of epitope residues are very similar to that of surface residues and far from that of interface residues. And this raised the following question. How come that an antibody that does have a unique amino acid composition itself binds specifically into a region that is basically not specific? So I will try to explain you now the solution we suggest for this question. So we can divide the epitope into residues that bind only the heavy chain or residues that bind the light chain. Now, if we will look on the amino acid composition of these residues, we will see that their amino acid composition is a little bit more different than that of surface residue. And if we will divide the epitope furthermore, and we look now on residues that bind one of the CDRs, we will see that their amino acid composition is even more different than that of surface residue. However, when you mix all the residues together, you get an amino acid composition that is almost identical to that of surface residues. So epitope residues are indistinguishable from surface residues. But why is that? And the answer is given in this figure. This is an example taken from a review we have published last year on antibody-antigen recognition. You can see here in the middle the structure of a lysozyme together with 3 of its many more antibodies in green, blue, and red. The residues on the lysozyme surface are colored according to the antibody they bind. You can see that almost any residue on the lysozyme surface is actually an epitope of a certain antibody. And this led us into our first conclusion, that an epitope should be predicted for a certain antibody and not for any antibody. But still, how would we do that? How would we recognize an indistinct patch? So maybe if we will understand how does the antibody bind and recognize its epitope, we will be able to use this information to predict it ourselves. So let's now see what happens in the antibody side. You can see here again 3 sets of amino acid compositions. In green is the amino acid composition of the paratope region of the germline antibody, which is the antibody sequence as encoded in our genome before the antibody meets the antigen in the first time. In red, you can see the amino acid composition of the somatic hypermutation introduced into the paratope region during the process of affinity maturation. This process happens after the antibody meets the antigen in the first time, where many mutations are introduced to improve the affinity of the antibody for its antigen. And in blue, you can see the amino acid composition of germinal protein-protein interfaces. You can see that each of these group of residues have a unique set of amino acid composition. Now, if we will look again on the measure, the measure of the difference between this set of amino acid compositions, we will see that the paratope of germline antibodies are quite different than the protein-protein interface. However, the amino acid composition of somatic hypermutations are more similar to protein-protein interface comparing with the original germline. And thus also the mature antibody, which is basically the combination of the germline antibody and the somatic hypermutations, is more similar to protein-protein interface than the original germline antibody. Or we can say in other words that the antibody is evolved during the affinity maturation to be more similar to protein-protein interface and to bind specifically its epitope. So now we can look not only on the amino acid in the antibody side or only on the amino acid in the antigen side, but also in the combination on amino acids from both sides, or what we refer to as the contact potential of the antibody-antigen interface. You can see here the contact potentials of the 6 CDRs, where in the x-axis you can see the amino acids from the antigen side, and in the y-axis the amino acids from the antibody side. And the cells are colored according to the preference of these 2 amino acids to be together in contact in the interface. You can see that each of the CDRs have a unique set of preferences. And this is what we believe the main point is. The epitope is indistinct. However, the interface and the combination of amino acids from both sides together are unique and specific. And what we need to do now is to use this information to predict specific epitopes. So this is our main hypothesis. We should predict epitope for a certain antibody, and we should do that using the antibody sequence. Based on this idea, we have developed PEASE, which is a fully automated tool that, given antibody sequence and antigen structure, predicts the antibody epitopes. Briefly, for each residue, a pair of residues, one from the antibody CDRs and one from the antigen structure, or antigen surface actually, these two residues are subjected into a machine learning model, which was trained over known complexes of antibody-antigen structures. The property used to train this model were calculated based on the antibody sequence, based on the antigen structure, and based on the contact potential. Each such pair of residues is assigned a score by the model, and this score is later translated into a residue score, which is assigned to each residue on the antigen surface and corresponds to the chance of this residue to be in the epitope. Finally, there is an optional stage of clustering predicted residues over the antigen 3D structure to generate predicted patches. So I will show you now a case study for which we applied PEASE. So the vaccinia virus is the active component of the smallpox vaccine, which is the first vaccine that was completely eradicated a human disease. Although this vaccine was used already many years ago, we still don't know what epitopes made this vaccine to be so efficient. D8 is the protein on the vaccinia virus surface. You can see its structure here. And it was suggested to play a key role in the efficiency of the vaccine. However, no epitopes were known on this protein before we started our study. We have received from our collaborators a set of 12 sequences of antibodies, all of them binding D8, and we were asked to predict the epitope of these antibodies over the D8 surface. Our collaborators also performed a cross-blocking assay of the 12 antibodies against each other. And you can see the results of this assay here, where black cells correspond to antibodies that completely blocked each other's binding to D8, and thus their epitopes are assumed to be highly similar or identical. Gray cells correspond to antibodies that partially blocked each other's binding to D8, and thus their epitopes are assumed to be partially overlapping or close in space. And empty cells correspond to antibodies that could bind D8 simultaneously, and thus their epitopes are assumed to be not close over the antigen 3D structure. Now, we use this information to cluster the 12 antibodies into 4 groups, where all antibodies of the same group are assumed to share a highly similar epitope and have the same relationship with all other groups. Now, we use this information to improve our predictions, and we did that in 2 levels. In the first level, instead of predicting a single epitope, a single patch for each antibody, we predicted a single patch for each group of antibodies, using information from all antibodies of the group. In the second level, we have drawn this scheme of the relationship between the epitopes, where we hypothesized that the epitope of group 1 should not overlap with any of the other epitopes. The epitope of group 3 should be partially overlapped with the epitopes of group 2 and 4, and the epitopes of group 2 and 4 should not overlap themselves. Then solutions or predictions that were not in agreement with this scheme were excluded. We have ended up with 4 predicted epitopes, where the patches predicted for group 2 and 3 antibodies were basically the same. Now, our collaborators made many efforts to experimentally map the epitopes of these antibodies, and they could do it very nicely. So for the antibody of group 1, they performed a deuterium exchange assay, and they could have identified 2 linear peptides that comes together on the antigen 3D structure. For the antibodies of group 2 and 3, they have performed an ELISA assay of peptides in length of 20 amino acids each from the D8 structure, and they could identify one such peptide that bind all antibodies of these groups. And for one of the antibodies of group 4, they could crystallize it together with the D8 protein, as you can see here. So we use this experimental mapping to evaluate our prediction. So residues that were experimentally shown to be in the epitope are colored, and predicted residues are shown in bold. You can see that all 5 predicted residues for group 1 epitope were indeed in the epitope. 4 of the 5 predicted residues for group 2 and 3 were indeed in the epitope, and 2 of the 4 predicted residues of group 4 were indeed in the epitope. We have also evaluated our predictions quantitatively, and you can see that the precision of the predictions was raised from almost 40 to almost 80% based on the incorporation of the data from the cross-blocking assay. So PEASE is freely available on the web, and you are welcome to try it. Let me just summarize. We have shown that antibodies evolved to bind indistinct patch on the antigen surface, and thus, in order to predict their epitopes, we need to use information found in the antibody sequence, and that using simple assays, we can improve the prediction significantly. And I would like to thank my supervisor, Dr. Yanair Fran, Anat Borkovich, who did the analysis of the somatic hypermutations, Vered Kunik, who did the analysis of the indistinguishability of residues and the contact potential, Guy Nimrod, who started the development of PEASE, Shauli Ashkenazi, who helped in the design of the PEASE server, and to Bjorn Peters, who led the collaboration that I mentioned, and all the people written here that were a part of this collaboration, and to the NSF, who funded my travel into here from Israel. And thank you.
So great talk. So in the prediction, you say use the sequence of the antibody. Does it use the CDR part or the hosting?
Yeah. So we used a tool was developed also in our lab for predicting the antibody CDR, so what we refer to as ABRs. This can predict it from sequence. And yes, we consider only residues from the antibody CDRs and residues from the antigen surface, right?
Okay, thank you.
It's an interesting talk. I want to know, when we talk about epitope, we almost consider the paratope because they are a pair all the time, right? So here you divide your antibody into 12, right? How can you divide?
Excuse me, how can what?
Yeah, you grouped your antibody, your paratope kind of, into 4 groups.
Yeah.
So what's the criteria? Because sometimes it's difficult.
Okay, so let me get back to this slide. You mean this, right?
Yes.
This one, right?
Yeah.
Okay, so based on the cross-blocking assay, that means that if antibodies were completely blocked each other's ability to bind the antigen at the same time, we assume that their epitope is the same. And then what we do, instead of use the information only from one antibody, we use sort of the average information found in all antibodies that compose this group. So it's some of manipulation of averaging the information from all antibodies of the group.
So actually, you didn't consider actually the exact, paratope.
So
yes, I know. So for this 12, so exact composition or a residue layout in the paratope? Did you compare them?
So the way it is done, I will try to explain it briefly. So it's first predicted for each antibody separately, and then the scores are considered together in some way to predict an average epitope. But for the first stage, the paratope of each of the antibody of the group is considered. We can talk about it later and explain you in more details.
Okay, yeah, another is just a comment. I haven't seen any, comparison between yours and the others.
Okay.
Because
so
now CEPA already published the version 2 online. Yeah.
Yeah, sure.
Okay,
so
thanks.
you can see this comparison in the paper that in the paper itself. It was published in Structure a few months ago, and you have also the reference here. But basically, we have compared to EPSVR, which was shown also in the paper that the review I've mentioned in the beginning, and in some other papers to be the most successful, or at least one of the most successful methods. So we have compared into it, but I have to say that we have to note that these two methods are not exactly the same, so the comparison is somewhat limited because they predict epitope for any antibody, and we predict epitope for a different epitope for each antibody. So we can do the comparison, and we did it, and we have published a precision recall curve, and you are welcome to see it in the paper. But you have to take it into account that it's not exactly the same objective of the method.
Hi.
Hi.
Nice talk. I'm not very familiar with this entire field, so I guess it's probably a naive question. So from this figure, you can tell that there's a lot of experimental evidence that we need to kind of tie these two pieces together, particularly this cross-blocking assay, right? But it feels like if we have enough of these out there, and I don't know how much data there is available, we could start talking about using these type of data to start talking about can we synthetically engineer antibodies to target specific epitopes using enough of these types of data? Are you guys thinking about those things, or am I just out?
Can you say thinking about what?
So instead of having, purifying these 12 antibodies, bleeding however animals you might have needed to bleed to get these antibodies and do this cross-blocking assay, given enough data of this type where we have several different antibodies targeting a particular epitope, we don't know exactly what's happening, but given enough of this data, it seems like we could start talking about how to identifying an epitope we want to target, and then synthetically engineering an antibody to target that instead of the long process of the bleeding animals that we're doing now.
So as I mentioned in the beginning, this is why one of the reasons why epitope prediction is so important, because if we will know the epitope, and this is one way to get into it, but if we know the epitope, then it should help us in the antibody design and engineering and so on, as you can say. So of course it's a stage in order to do the other stuff, and it brings us.
Is there enough data of this type out there where we can just start kicking the tires on some machine learning type of approach that might be able to do that? Again, naive question. I guess that's just.
Yeah, it really depends. For some cases, we are able to get to better results. In some cases, not so good. But I can definitely say that if we can perform such assays, such as cross-blocking data, then we can significantly get into much more accurate results. So.
Can your method incorporate information about the antigen? For instance, if you know that a specific region is blocked by glycosylation, can your method distinguish and deal with that?
We didn't take glycosylation into account, but if you have any information on which specific residues that should not or should be in the epitope based on mutational data or things like that. So you can definitely incorporate it in a post-processing way. And like we use this information to exclude solutions that were not in agreement with this scheme, we can also exclude other solutions that are not in agreement in some other experimental information, such as mutations and so on. And then I believe we can get to better predictions.
Just a quick question. So a very interesting talk. I really enjoyed. So my question is, have you tried to expand this to a bispecific antibody or some?
I couldn't hear. Can you please repeat?
Have you applied your method to a bispecific antibody or some peptide? You know, this kind
Did we applied it to a specific?
It's very popular.
Not yet, but not yet. But I think it's a very interesting idea. We can try it. Yeah.
Thanks. You know, even the peptides or something. Those things to me are very.
Maybe we can talk about it because it's beginning. Thank you.